All right. Welcome, everyone. I feel like every lecture I give, I'm telling you, oh, this is my favorite lecture, but this is my favorite lecture. I also heard that some people change their projects to work on 3D structure, after 3D genome architecture after the 3D genome lecture. So I hope you guys don't keep changing topics every lecture, because there's just so much cool material coming up. Today we're going to be talking about regulatory genomics. So number one, how do we discover regulatory motifs? How do we find their instances and use that to predict their targets? And then how do we dissect regulatory regions within which these motifs are found? So we are now in the first lecture of the third module. So we talked about aligning and modeling genomes. Module 2 was all about gene expression and epigenomics. And we're now getting into the circuitry. So today we're introducing the foundation of the regulatory networks inside the cell by learning about regulatory motifs, and then how do we discover, present them, and find their targets. And then on Tuesday next week, we're going to be talking about network structure. And on Thursday next week, we're going to be talking about deep learning. So the goal for today is, number one, introduce the basis of gene regulation in most genomes, focusing on mammalian genomes. Number two, introduce computational techniques for discovering these motifs based on expectation maximization and Gibbs sampling. Number three, introduce computational techniques for finding the instances of these motifs. So a motif is a construct that spans across many different regions and summarizes them. And then this effectively generative model of these sequence patterns can instantiate in different locations in the genome. And part 4 is going to be about finding these instances. Actually, number 5 is going to be finding these instances. And number 4 is going to be about exploiting many of these instances together, regardless of who the regulator is, to learn regulatory motifs de novo. So basically an experimental driven technique and evolutionarily driven. And then number 6, we're going to be talking about how do we discover these motifs in larger regions? How do we use experimental techniques in extremely high throughput for dissecting the functional nucleotides within larger regions? Ready with me? All right. So what are these regulatory motifs? I showed you on the very first lecture a region of the yeast genome where I highlighted nucleotides that were important in red. And these were instances for the GAL4 and the MIG1 regulators, as well as for the data binding protein, TBP. So how do these motifs work? So these motifs are basically constructing a landing pad for different regulators, for different transcription factors. And these landing pads are made out of atoms that are just in the right locations, as well as the entire double helical structure of the surrounding region that the protein then recognizes. So that protein makes contact with the phosphate backbone of DNA, and this does not depend on the specific nucleotides under this backbone, but it also makes contact with the stacked nucleotide base pairs of the DNA that it senses from the side. And depending on what nucleotide you put at a particular position, different atoms are going to be facing that protein, and then they will be making a contact or not making a contact. Who's with me on this one? Raise your hands. Who feels that they actually learned something in the last minute? No, I'm serious, because this is not obvious. Basically, everybody thinks about, oh, we're just going to recognize that motif. But that motif is not recognized by opening up DNA and then reading the bases in the same way that RNA is made out of DNA. That motif is recognized in the closed double helix, not by the traditional ACGT base pairs of 3 hydrogen atoms, but all of the other auxiliary atoms that are on the side. This is, in fact, what leads to the degeneracy of regulatory motifs. Sometimes the specific molecules, the specific atoms that the protein binding recognizes can be achieved by a C or a G at that position. an A or a G at that position, and so on and so forth. And that would allows these motifs to be degenerate. That would lead to these, you know, W, which means weak, which means A or T nucleotide. Thank you so much. Does that make sense? So basically, regulatory motifs are turned on or off. Basically, genes are turned on or off in response to changing environments. And as I mentioned previously, there's no direct addressing. There's no go to chromosome 13 at position 327,542. Instead, the subroutines, quote unquote, the genes, contain sequence tags, i.e. motifs, and then there's specialized proteins, i.e. transcription factors, that recognize these tags. So I'm going to put this red tag in front of all of the genes that I need for metabolizing galactose, and I'm going to put this blue tag in front of all the genes that I need for, you know, that I don't need when I metabolize glucose. So therefore, if I sense galactose, Gal4 goes and binds, and it turns on all these galactose genes. But if I sense glucose, which is a more efficient sugar for yeast, MIG1 is going to turn on and then shut off these genes, and then other genes will turn on. Okay? So what makes motif discovery hard is that motifs are very short. They're only 6 to 8 base pairs long, and they're sometimes degenerate. As I mentioned here, you know, you can have any position being a degenerate position. They can contain distances between them. In this particular case, the Gal4 transcription factor binds on the same side of the double helix, but 11 nucleotides apart. Every 10.5 nucleotides, I have a full turn of the double helix. That basically means that if I bind on the same side, I'm going to bind 2 parts of a motif, which are going to be spaced by about 11 nucleotides. Okay? And then the motifs can act at variable distances, both upstream and downstream of their target gene. Who's with me so far? Any questions? You really did not learn anything? Who feels that they've learned something? Let's make him happy. He seems to care. All right. So the regulatory code is all about finding where are these motifs and how do they function, and where are the corresponding regulatory regions that they define and how do they function. And motifs are everywhere. Motifs can be right where the transcription start site is, recruiting the specific location where RNA polymerase will start transcribing DNA into mRNA. They can be in the boundaries between different exons and introns, basically enabling the splicing regulators, many of which are RNA binding proteins or RNA binding RNAs, to basically bind and guide splicing. Some of them are post-transcriptional, and they guide the binding of microRNAs, which we talked about last time. And some of them are acting at very, very large distances. Some can act at millions or more nucleotides away by sort of creating this looping interaction between this distal enhancer region that will then turn around and find these target promoter regions. Okay? So this is the part space. Basically, there's 20 to 30,000 genes, and then the circuitry is all of these regions: enhancers, promoters, splicing, and, you know, post-transcriptional motifs. And then the regulatory code has basically some kind of combinatorial coding of unique tags. So, you know, we use multiple individual motifs, and together they allow this data-centric encoding of these addresses. And these are overlaid with memory marks. And then these are the epigenomic states that we learned about last time. And all of this allows us to basically modulate this, you know, large-scale, you know, encoding of regulatory information. Okay? So, as I mentioned, transcription factors basically use DNA binding domains to recognize specific DNA sequences in the genome. And here you can see one such example. And you can see how this is the DNA molecule. As I mentioned, it's a very cool molecule. It's got a major group. It also has a minor group. And in that major group, in this particular case, you basically have this protein that's sort of coming here, and it's making specific contacts with specific positions. So you can see here, these amino acids of that protein are contacting, you know, site far away. These ones are sort of forming many, many different interactions in this loop. And then, you know, the other one comes, loops around, and then contacts another. Okay? Everybody has an intuition now for how these motifs work? Yeah. Correct. Other questions? So that's one motif. There's another motif. There's another motif. Notice motifs have some very cool properties. This particular motif, if I read it backwards, what does it spell? Come on, can you read it backwards? T-A-A-T-T-A. Wait, I asked you to read it backwards. This is forward. My son would have such a blast on this. He's 6 years old, so he could spend all afternoon saying, hey, wait, I told you to read it backwards. So anyway, it's a palindromic motif. It's actually a reverse palindrome. So the complementary strand reads exactly the same as the forward. Can you read this one backwards? I told you to read it forward. Come on. All right, so anyway, this is a palindrome as well. So many motifs are palindromes. Why? Because they don't really care. They can just bind this way or that way, and it's just as good. There's no directionality in their binding. It doesn't matter how they bind. Others are not palindromes. This is not a palindrome. So this one has the possibility of caring about which direction it binds in. It may have a different effect, one direction or the other direction. We saw for the CTCF regulator that it is in fact extremely directional. In one way, it stops as a stop sign. Cohesin cannot go through. On the other way, cohesin goes through no problem. Okay? So directionality actually matters. And, you know, the specific structure of the motif dictates how things are going to bind. And these motifs are so important because if we disrupt nucleotides from, just the natural polymorphism that exists within our DNA sequence from human to human, we can actually disrupt functional nucleotides and then lead to a phenotypic difference and sometimes disease from person to person. Not because of protein coding variation, but because of non-coding variation. That's why it's extremely important to sort of systematically understand the circuitry of the genome. Okay? Everybody with me? So, quick question. What fraction of genetic variants perturb a protein directly? If you look at all of the common variation that has been studied with genome-wide association studies, what fraction of these regions disrupt a protein? Who wants to take a guess? Raise your hands, just take a guess. What fraction disrupts protein? Yeah. So 1% of the human genome encodes protein. And therefore, you would expect that if polymorphisms are laid out by chance in the genome, randomly, uniformly, you'd expect that about 1% disrupts proteins. But the question is, for those that are associated with disease, not just any genetic variant, but the disease-associated genetic variants, what fraction of those disrupt proteins? Yeah. 15% or 50%? 15, good. Anybody else want to say higher or lower? Higher. What fraction do you think? More than 50%. So you would expect that proteins are just so darn important. Half the time, these common variants are going to be disrupting a protein. Turns out you're absolutely right. Proteins are so darn important, but she's not quite right enough. It's actually much less than 50%. It's even much less than 13%. It's only about 7% of the time that genetic variants actually disrupt a protein. And the reason lies within his insight that proteins are so darn important. That if you disrupt the protein, that genetic variant is not going to be common enough in the population because of selection. So genetic variants that disrupt proteins are in fact not as common as genetic variants that disrupt the rest. And the rest is effectively 93% of cases. These genetic variants actually disrupt a motif or some kind of non-coding function rather than a protein direct. Okay? So today's lecture is kind of important. If you care about the 93%, you don't focus on the 7%. All right. So as I mentioned earlier, we can actually identify individual nucleotides that can, yeah. Yeah. So you have a very good point. Is there a one-to-one correspondence between amino acids and the nucleotides they contact? And the answer is absolutely not. If you look here, how many nucleotides there are? 1, 2, 3, 4, 5, 6, 7 base pairs. Okay? And these two are contacting the same one. These three are contacting the same one. This one is not contacting a nucleotide at all, and so on and so forth. So the answer is, we wish it was that simple, but it's actually ridiculously hard. There are some proteins called zinc finger proteins, where there's more of a code, where every 3 amino acids sort of contact one nucleotide, and therefore you can actually design them to sort of construct new motifs. But most of the time, it's just some crazy combination that has been evolved, not engineered. All right, so 93% of cases, this is what's happening. And there's one very beautiful example that Melina Klausnitzer worked out, working in collaboration with my lab, who basically showed that you can actually trace down the strongest genetic association with obesity down to a single nucleotide by disrupting this T nucleotide and changing it to a C. This AT-rich interaction domain can no longer bind there. Then that leads to de-repression of a super enhancer, a very massive regulatory region, de-repression of these two genes, 1 million nucleotides away, and then a switch from energy burning of thermogenesis to energy storing of adipocytes, lipid storage. And all of that explains 7 pounds in a human adult. So basically, those who carry 2 risk versions for both mom and dad for this particular region, thank you, mom, thank you, dad, I got both, have 7 more pounds as an adult than, otherwise. So when I go to the doctor, I can say, hey, I'm actually 7 pounds leaner. It's just mom and dad gave me this version here. Anyway, it's not deterministic, and human brain has something to do with it as well. But anyway, this basically changes your metabolism due to a single nucleotide change in a regulatory motif that's this small, that sits 1 million nucleotides away from the target gene. And then this is sort of different types of structures for how they bind. They can basically bind, within the major groove. They can bind across different regions, across different sides of the helix. They can, bind very often. When you get binding as a homodimer, for example, GAL4 is a homodimer. One arm contacts CGG on one side of the helix, and the other arm contacts CGG on the other side of the helix. So, instead of nature basically making 2 different elements that will both recognize the same motif, it just makes one and uses it twice. And then that's why a lot of motifs are palindromes, because you can actually just very easily put together 2 of them. Okay? So the proteins feel DNA. The 3D topology dictates the specificity. And this is not the only type of recognition. MicroRNAs, for example, actually bind by complementarity. Nucleosomes care a lot about GC content. And then various RNAs can bind using both structure and sequence. Okay? So given this very complex structure, we'd like to have a representation for capturing what this language of gene regulation looks like. And this representation is very frequently position weight matrices. This is a matrix that basically at every nucleotide position gives a weight for each of the 4 nucleotides. Here's one such example. This is the motif for the ABF1 protein in yeast. And this is the consensus. So this position is nearly always a T. That position is always a C. Always an A, always an A, always a C or a G. And here, half the time it's an A, half the time it's a G. Similarly here, half the time it's an A, half the time it's a G. Half the time it's a C, half the time it is a T. And you can see here from all of the instances of that motif across the genome, you can basically find all places where the ABF1 regulator binds. These are all the target genes. And then these are the coordinates upstream of the transcriptional start site where they bind. And then this is the alignment of all of the motifs. And then if you count at every position, how frequently did I see each of the nucleotides across these hundreds of sites, you end up with exactly this. Who's 100% with me here? Awesome. So what is the limitation of this representation? What does it assume? Correct, correct. So they assume that only the nucleotides matter, and they don't deal with shape information or folding or other kinds of information. That's good. What else does this representation not capture? Yeah. Good. So it basically says that every position is equally important. It only tells me the specificity of that position, but even if this is always an A, maybe it's not as important. So it doesn't weigh them by importance. So I could have an overlay of this representation that also weighs each of them by their importance. What else is? Yeah. Yeah.
It doesn't account for combinations. So maybe the answer like always for A or always for B, but like you cannot really know.
Correct. It basically assumes that every position is independent. It basically says that, you know, whether I have an A or a G at this position, doesn't really matter if I have an A or G at that position. But maybe in practice, every time I see an A here, I always see an A here and a C here. And every time I see a G, I see a T and a G. But then they don't go, they don't mix and match. Everybody sees that? Raise your hands if you see that. Great. So basically, PWMs assume independence. And that's okay for most of the time. For some of the time, it's not okay. And when it's not okay, what we can do is expand the PWM into multiple PWMs, multiple position weight matrices. And basically say A, TCA, C or T, blah, 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 ACG, A. It would be one of them. And then G, the same thing. G would be the other one. And then suddenly you've captured this dependency by splitting the motif into two, for example. Or you could have more complex representation that capture these dependencies together. Some people just use clouds of k-mers. They basically say, here's all the k-mers. I'll just use a cloud of them, you know, mixed together somewhere. Everybody with me so far? Awesome. So they assume independence, fixed spacing. For example, you know, this representation will not be able to capture a variable spacing. And you saw earlier how a protein can bind two different locations. And maybe the spacing doesn't really matter. It just looks for these two halves. And this representation doesn't capture spacing. Yeah, so we make the simplifying assumption that if I score this sequence using this, that the score that I would obtain is an approximation of the affinity. But you're right, the two are not exactly the same. Yeah, yeah. Correct, correct. Basically, we assume that there are enough of these samples and that they are representative of the true, you know, frequency of binding. And then from affinity, from frequency, we basically, you know, infer an approximation to affinity. Okay? Great. And this is, you know, there are many experimental ways of finding this affinity. What I mentioned earlier is that, you know, experiments have basically been done to see where is that transcription factor bound. For example, using a chromatin immunoprecipitation experiment, you can basically say, oh, this regulator ABF1 is sitting exactly here on the sequence. Therefore, I'm going to go and cut that sequence out and then figure out what's there. So there's something called footprinting, which basically allows you to sort of digest the bound protein and DNA complex, digest the DNA away, and then whatever does not get digested is where the transcription factor was sitting. So you can use that to basically find where was it sitting each time, but that doesn't give you the motif. That only gives you the precise coordinates of binding. To get the motif, you need many locations. And that's sort of the key thing to recognize, that motifs, in fact, capture information that's shared across many positions, as opposed to an individual instance, which is a fully defined sequence. And every motif can be thought of as a generative model. This is basically telling me how to generate more instances of the same class, just like an HMM that we talked about earlier. So here, I can sample from that probabilistic model. I can basically sample, and then, you know, half the time I'm going to get an A, so half the time I'm going to get a T, or a G, and so on and so forth for every position. Okay? Everybody with me? Great. So how do we find these motifs? So if I have a particular protein and I want to find out what it binds, what I could do is hybridize that protein to an array that contains many, many different versions of nucleotide sequences. And then I can just wash that protein through that array. And then I could basically find all the DNA spots that light up. And that can basically tell me the affinity of that protein has something to do with all of these k-mers. Then I can analyze all these k-mers together and then infer a motif from that. Or I could basically do DNA precipitation with microRNA detection, basically, pull down my protein and then do a relative hybridization in, either genomic sequences or random sequences, then figure out where it hybridizes. Or I could actually evolutionarily, do rounds of selection where for every protein, I keep all of the DNA sequences that it recognizes, and then I, keep filtering until I find all the true DNA matches to that particular protein. Or conversely, I can use DNA to select proteins that bind it, and then do multiple rounds of selection, and then find the best protein match to a particular DNA sequence. So, computationally, there are many ways of dealing with motif discovery. So these are some experimental methods. Computationally, you can basically use expectation maximization with Gibbs sampling that we're going to look at today to find motifs that are overrepresented in a set of sequences. Or you could enumerate all possible motif sequences with wildcards. Or you could basically look at the correlation between the particular motif and the positioning within a ChIP-seq peak where this motif occurs. And the closer you get to the center of the peak, the higher your score. So that can actually allow you to use a cutoff-free approach rather than just saying, oh, it's between here and here. It allows you to do a much smoother approach. So these are all based on a particular set of sequences that contain that motif. You could also use genome-wide conservation by basically looking at not just one instance of this CGG spaced by 11 of CCG motif, but the entire genome and all of the instances to basically say how frequently are they conserved. And you could also deal with specific protein domains using PBMs or selects, as we saw in the previous slide. And then again, motifs are not limited to DNA sequences. They can be splicing sequences. They can be protein sequences. It could be recurrent patterns at the physiological level, and so on. So I like to think of these challenges in regulatory genomics as basically graphs going from each of these entities to the other. So what are these entities? On one hand, you have the regulator. This can be a transcription factor or a microRNA or a protein binding RNA or an RNA binding protein and so on and so forth. These could be the motif, the specific sequence, and this can be at the DNA level or the RNA level or even at the protein level. And then the target, which basically tells you not just the general sequence specificity of a motif, but the specific instances where that actually occurs, the individual places in the genome. And then to recognize the TFs, we can basically look at, their homology, the homology of every protein to known transcription factors and known domains, basically discovering new transcription factors. We can use evolutionary signatures or experimental cloning to discover new microRNAs. To discover the motifs, we can use de novo discovery, as we're going to see at the end of the lecture. To discover the targets, we can use these evolutionary footprints or these actual DNA footprints. We can digest everything that's not bound by the factor, and then end up at these specific locations. And then, so this is basically techniques that only focus on the regulator, only focus on the motif, only focus on the target. But then to go from the regulator to the motif, if you have a particular protein that you've now discovered and you'd like to know what does it bind, that's where you could do a protein binding microarray experiment or a Gibbs-seq or a selects experiment or a ChIP-seq experiment, and then find all of the targets and then computationally find common sequence patterns in those targets. If you have the motif and you'd like to find the regulator, that's a little harder. You basically have to use the motif to pull down, using a piece of DNA, to pull down all the proteins that might be binding that piece of DNA, and then do a mass spectrometry experiment to basically figure out what are the corresponding proteins. If you have the regulator, you'd like to find the targets. A ChIP-seq experiment or ChIP-seq experiment will basically give you the targets directly. And then from the targets, you can infer the motif using enrichment. And then from the targets, you can find the regulator using, network analysis. And then from the motif, you can find the targets using evolutionary signatures. So basically, there's a lot of problems relating regulators to motifs to targets in any of these directions. And if you have one, you can find the other using this. So now, let's focus on exactly this arrow. I have a bunch of common targets of a particular protein. For example, I did a ChIP-seq experiment or a ChIP-seq experiment, and I found a bunch of intergenic regions that are all bound by the same protein. Alternatively, I may have just looked for a bunch of genes that are co-expressed. They turn on and they turn off at the same time. That suggests that they have some common regulation, and therefore they might contain a common motif. So let's now focus on how do we find an enriched motif in a set of either co-regulated genes or co-bound regions. Okay? Everybody with me on this slide? Awesome. Any questions? Yes. Exactly, correct. We have to find, number one, for every gene, what are the regions of regulation? And then from the regions, go to the motifs. You're absolutely right. So the epigenomics lecture can help you here. You can basically say, if I have a bunch of co-regulated genes, what are the putative regulatory regions? This is where I'm going to have to look. In yeast, these are very nearby to the gene. In, higher, more complex species, you have to search hard. Very good question. All right, so now let's look at this enrichment-based discovery. So given a set of co-regulated or functionally related genes or regions, can we find common motifs in their promoter regions? So basically I have a bunch of genes and I like to find common motifs within them. So what can I do? I can basically use sequence alignment to align the promoters to each other using some kind of local alignment technique. Expert knowledge for what the motifs might look like. I can find some kind of median string that basically tells me, by enumeration, either sampling the motif or sampling, every k-mer that I find in my sequences. And I could also start with evolutionarily conserved blocks in the upstream region. So, these are just some of the ideas of what I could do to find these common motifs. But fundamentally, what I have is a problem whereby if I knew the positions of those sequences, I would be back to my first slide, or all I would need to do is simply count at every location if I knew exactly where they start. If I knew their sequence pattern, then all I could do is just simply search that sequence pattern in each of the regions to figure out their starting position. But I don't know either their starting positions or the pattern. So what can I do when I want to know two things, neither of which I know, but that are dependent on each other? Expectation maximization. So I can basically iteratively assume I know one and then use it to find the other. Okay? So what I'm trying to do is effectively figure out, in these previously unaligned sequences, what are the starting and ending positions of that shared motif, and what is the specific profile. And what EM does, and all of my slides have been messed up for some reason, so all of these boxes have been resized. I fixed some of them right before the lecture, but not all of them. All right, so basically, given the profile matrix, it is easy to find the starting position probabilities by simply scoring every single position. And then given the starting positions, it is easy to infer the matrix. I just simply count, okay, at every position, how frequently did I see each of the bases. Raise your hands if you're with me on those two steps. Awesome. So that's effectively the expectation maximization procedure, okay? Given the motif, I can figure out the starting positions, and given the starting positions, I can figure out the motif. So the key idea is an iterative procedure for estimating both given the uncertainty. And then it is effectively a learning problem with a nuisance parameter, a hidden variable, which is the starting positions. And that's where we go back to this table that I showed you before. We've seen these before with HMM. We basically, the first time we saw it was hidden Markov model. We basically said, hey, we don't know the path, we don't know the annotations, and we don't know the parameters. So what we're going to do is assume some annotation, and then use that to infer the parameters, and then go back and forth between them. So basically, in the E step, we were assigning every instance to a particular label probabilistically, or assigning labels to points. So basically, this is fitting very nicely into that framework we saw before. And now in the Motif discovery instance, the hidden label will be where are the starting positions, or alternatively, what is the Motif definition. And then greedily, I could find the best Motif match in each sequence. And that's sort of probably the first thing you had in mind. Just, if I have a particular sequence pattern, I'm just going to search it and see where does it match. I'm going to use that. And that's effectively the equivalent of Viterbi training, or k-means. The expectation maximization version, which is implemented in the MEME software, in fact, does not say what is the best position. It instead says, can I have that Motif definition and search all possible positions and weigh each of them by their probability of matching that Motif. And then what Gibbs sampling does is that it only uses one position, but it picks that position at random, not completely at random weighted by the Motif match scores. Okay. So then the basic iterative approach is that I have some length parameter, I have some training set of sequences, I have some initial values for my Motif, and then I iteratively re-estimate the starting positions given the Motif, and then re-estimate the Motif from the starting position. And then when that stops changing, updating, because I found now a nearly optimal Motif, when the change is very small, I just return both the Motif and the starting position. So how do we represent that? Well, if we have a fixed length W, we can represent the Motif by a matrix of probabilities that basically tells you for every position, what is the probability of each character. And then this can be represented in a matrix. For example, if my Motif is more or less CAG, then the first position is mostly a C, second position is mostly an A, third position is mostly a G. But it's very probabilistic. So, CAG is not that far off from GAG in this particular Motif. And then that's my Motif model. I also need the background. And the background could be a near uniform background, which basically says every base is equally frequent. Or maybe in a very GC-rich genome, I'm going to have a much higher frequency of G and C. And then recognizing GC-rich motifs will be harder because they'll be very similar to the background. And an AT-rich motif with the same information content would score higher because it would be more dissimilar from the background. So that's for representing the motifs. How do we represent the starting positions? Well, the starting positions is simply a score at every nucleotide position that basically tells me what is the frequency, what is the probability with which the motif is starting at that position. And we can represent that in some matrix Zij that basically tells us for every sequence I and every position J, what is the probability of starting a motif instance at that location. Okay, although that should be quite trivial. So now, if I have my starting positions, Zij, which basically tells me the probability of starting at every location for every sequence. And if I have a motif matrix, which basically tells me what is the probability of seeing each of the characters in every position of the motif, then I can now define mathematically what does it mean to go between them back and forth. Okay? So computing Zij, the matrix of instance probabilities from the motif is very straightforward. At every position, I evaluate the start probability by simply multiplying across the matrix. If I have, G, G, A, C, T, I go G, G, A, C, T, and I just multiply it across. Okay? So that basically tells me the probability of the motif matching, but what I want is a likelihood ratio. So I also want to multiply that with a background model, which can be a one nucleotide background model or dinucleotide or trinucleotide background model. And then I'm going to score that mononucleotide or di or tri at every starting position. And then I'm going to come back with a likelihood ratio. How much more likely is it that this particular sequence was generated by the motif rather than by my background model? Raise your hands if you're with me. Awesome. Great. Any questions? Okay. So, and now, regardless of what the motif looks like, I can score my Zij in this particular fashion. But once I've scored it, how do I select the locations from which I'm going to rebuild my motif? Well, I can use expectation maximization, where I choose all of the starts weighted by their distribution. I can use Gibbs sampling, where I pick a single start for each sequence by sampling from that Zij. Or I can use a greedy approach, what is the best starting position for every sequence, and then only choose that one. So how do we now calculate Gij? So I basically have my probability of starting at any particular position given my sequence and given my motif model. And then this is the posterior probability of that starting at that position. But what I have is a generative model that basically tells me what is the probability of emitting a particular character given my motif at any one position. So what I'm going to do is use Bayes' rule, just like we have before. Given the prior of having a motif start at that position, given the evidence of what is the probability with which my generative model can generate the entire sequence, and then given this generative model, I can infer the posterior probability. Okay. At every iteration, I can calculate Zij based on the previous motif and the previous, definition, and then we can calculate that very easily. And then to obtain the total probability, we sum over all starting positions. This P of X is simply, the total probability, and then we assume uniform priors, or we could assume some more interesting priors. For example, I can look at evolutionary conservation and look at the frequency with which every nucleotide is conserved, and then maybe use that as a prior, that maybe there's a motif that's more likely to start there given the conservation. So if I'm scoring this particular motif, which as we mentioned earlier is GCT at every position, then at the first position, I have a high score that's obtained by 0.3, 0.2, 0.1, and then the rest is by background. At the second position, it's CTG, so I read CTG and I multiply across, so 0.4, 0.2, 0.6. And then at the first position, I generate it from the background, and all the other positions are generated by the background. Who's with me so far? Great. Then I can do the same at CTG and so on and so forth. Okay. So then that gives me, the Z vector. And then the denominator is simply P of Xi, which is, when the motif position is known, I can just simply calculate all of the scores before the motif, all of the scores after the motif using the background model in each case. And then for every position within the motif of length W, I basically have, the particular scoring by reading off the matrix. Okay. So I can basically simplify this by simply having the exact same score before and after. I can combine all of that together and then simply score the motif itself. So I can speed up my computation by only computing that and assuming that the rest will be the same at every location. So that's the E step of basically creating the Zij vector from the motif matrix. What about the M step of inferring the motif from those positions? As I mentioned earlier, there are three different approaches: the greedy approach, the Gibbs sampling approach, and the expectation maximization approach. Gibbs sampling only picks one at random. Greedy always picks the maximum. And then expectation maximization averages everything weighted according to their probability. So in this particular example, greedy will always pick the maximum. In this particular case, all methods more or less agree because there's really only one maximum. But in this particular case, EM will basically use both of these to estimate. Gibbs sampling will only pick one or the other at random. It'll pick one with this one with 2/3 probability, for example, and then we'll only use that one. And we'll pick this one with 1/3 probability, and then we'll only use that one, even though it was a lower probability one. And then greedy will always pick the max. And in this particular case, greedy will ignore most of the probability mass. Gibbs sampling will rapidly converge to some choice, and EM will basically average over the entire sequence together. Who can see the difference between Gibbs, EM, and greedy? Raise your hands. Awesome. Great. All right, now given this annotation, we can basically estimate the motif using the exact count divided by the total number of times I saw this particular sequence. And then I can add some pseudo counts each time to avoid overfitting. And then I can, from this instance and that instance together, I can estimate the probability of my motif. I can add some pseudo counts and then end up with, that motif definition, either using, an average of all of them weighted according to the probability, sampling one of them, or selecting the next. Okay? So what EM will do is iteratively refine the motif, the starting positions, the motif, the starting positions, the motif, the starting positions, converging in a provable fashion, always in some local maximum, but not always in a global maximum. So to do that, we basically initialize at multiple locations and then hope that one of them will actually converge all the way to the true maximum. Okay? Who's with me on EM and Gibbs sampling and all of that? Great. So basically, this was, going from the motif to the positions and back and forth. So I'm going to probably just, skip this section. We talked about that already. And now switch gears. So we basically now know how to find a set of common motifs from a set of sequences that were co-regulated or co-expressed or co-bound. What we can do now is look at a completely orthogonal approach for discovering motifs. And what this is looking at is a de novo approach, where we don't need to know the transcription factor in advance. So basically, every region-centric approach needs to have a set of regions that were either bound by the same transcription factor or co-expressed, co-activated in the same condition, and so on and so forth. But these TF-centric approaches require knowing the transcription factor or some antibody to a factor, and they take a lot of time and money to basically generate an experiment for every single regulator. There are thousands of transcription regulators in human. Not every one of them has been profiled experimentally. And what the novo discovery tries to do is effectively use evolutionary conservation in a completely unbiased fashion to basically discover a catalog of regulatory motifs without actually knowing what transcription factor this comes from. So then the key insight is that if I look in a particular location where this factor is bound, I will find one instance of that motif and it will be evolutionarily conserved, but so will the surrounding bases. And then if I look at many, many instances, we could, join them together across the genome. So basically what we're going to do is we're going to increase power by testing the conservation in many regions. So if I have CGG, CCG, CGG, CCG, CGG, CCG, in many, many different places across the genome, together they will sort of gain more power. So then the idea is that I'm going to be testing genome-wide conservation, taking all instances of a particular k-mer, and then asking how often is that instance conserved. And then for those k-mers that are conserved more than others, I'm just simply going to get a higher score, a higher genome-wide conservation score, and then they will perhaps be more likely to be regulatory motifs. Okay? So then if we try this with the GAL4 example that I mentioned earlier, so CGG, spaced by 11 nucleotides from CCG, we can now start measuring each of these scores. Yeah. That's exactly right. So basically the key intuition is that in one instance, evolution basically tells us that this motif seems to matter more because 3 of its instances are conserved in this particular region. And if we extrapolate to the whole genome, if it is preferentially conserved at every single location, even if it's not always perfectly conserved, it just needs to be preferentially conserved, together that signal will amplify. Does that answer your question? Correct. So we're going to be looking outside the genes. What's really interesting, and I'm going to get back to your question in a second, but basically what's really interesting is that regulatory motifs within genes are actually less conserved than random nucleotides. Evolution is actually somehow disfavoring motifs that occur within the gene. And you can see why. Because a transcription factor will suddenly bind there and maybe prevent transcription or interact in negative ways with the stuff surrounding. So if we do that for exactly the Gal4 motif, CGG, CCG, spaced by 11 nucleotides, we find that in all intergenic regions, it is conserved 13% of the time, compared to only 2% expected using random k-mer controls. If we ask what is the ratio of intergenic conservation to coding conservation, we find that Gal4 is preferentially conserved in intergenic regions versus coding regions, whereas random controls are, of course, preferentially conserved in coding regions versus intergenic regions. And you can see here that not only is it more conserved in intergenic, but it is actually less conserved in coding regions. And then lastly, you can test about upstream versus downstream. So basically in yeast, genes are very close to each other. You can ask for regulatory regions that are upstream of both flanking genes and intergenic regions that are downstream of both flanking genes. And what we find is that Gal4 is preferentially conserved in these divergent regions, but almost never conserved in these convergent regions, whereas random k-mers actually have the same evolutionary conservation. So that basically gives us evolutionary signatures for discovering these motifs. I can basically simply enumerate k-mers, and according to each of the signatures, basically find k-mers that are highly conserved in intergenic regions, k-mers that are preferentially conserved in intergenic versus coding, and k-mers that are preferentially conserved upstream versus downstream. You have a question? All right. So let's try that. We're just going to simply enumerate k-mers. So this is what we find. If we do test 1 of intergenic conservation and we ask for every k-mer, which is a dot here, how often does it appear and how often is it conserved? We find that there's quite a strong diagonal here with most motifs or most k-mers being conserved at roughly the same rate. The more you have instances, the more instances you will have conserved. But there are some k-mers, shown in blue here, that are much more conserved than what you would expect based on their number of instances. And one of them is, in fact, the Gal4 motif, CGG, spaced by 11 from CCG. But there are many, many more. So I can simply use this biological insight about the preferential conservation of evolutionary of true regulatory motifs to enumerate every k-mer, and then out will pop things that are preferentially conserved that are more likely to be motifs. Who thinks this is kind of cool? Awesome. So I did not need to know what the transcription factor is. I just simply said, what are the k-mers that preferentially conserved? Let's try the intergenic versus coding. Yeah. I'm just simply counting how many times is CGG, spaced by 11 nucleotides, from CCG occurring in the yeast genome. And I find that it occurs about 300 times, of which about 50 are conserved. That's a gap. So basically, this CGG-CCG pattern is what I have here. CGG doesn't matter what's in the middle from CCG. Does that make sense? So what I'm searching for conservation is only these 6 positions. The middle positions I don't care about. So that basically means that I search for CGG, CCG in the yeast genome in Saccharomyces cerevisiae, and then I ask Is it conserved in that other species? Is it conserved in that other species? Is that conserved in that other species? And I find that only a small fraction of them are actually conserved in the other species. Does that answer your question? Great. Correct. Yeah, so here I have Saccharomyces cerevisiae, Paradoxus, Micatai, and Baianus. And then for every single instance, I will search all red instances, and I will count how many of them are actually conserved. So I have a whole genome alignment of all these species. Therefore, I can simply search for how many are conserved. Yeah. Yeah. So we're going to have a lecture on comparative genomics, and there we're going to talk about how branch length matters. Why? Because if I only have, basically, if I align 1,000 Saccharomyces cerevisiae species, any one position has only one chance in 1,000 of being different from each other. And then if I pile them all up, every position will have some small chance of being mutated. If I take much more distantly related species, then every position has many more chances of being mutated. It's a question of power. The more branch length I have, the more power. Therefore, taking different species gives me a lot of branch length for relatively few sequencing experiments. I could do the same with maybe 1,000,000 yeast species or maybe 1,000 yeast species, but it really depends on how divergent they are from each other versus how divergent the species are from each other. There's a whole slide on that in the comparative genomics section. All right, everybody with me on the first slide? Great, yeah. The gray dots. So basically, all the dots should be initially gray, but afterwards I asked, how unlikely is it to find that many conserved ones, given how many I had observed? And that gave me a probability of being over-conserved, and these points are colored blue. Does that make sense? All right, so we can do the same for intergenic versus coding. What is the coding conservation? What is the intergenic conservation? And then what we find is that most k-mers have more conservation in genes, as you would expect. But there's some k-mers that are preferentially conserved in intergenic regions. Maybe these are my motifs. And then when I looked upstream versus downstream, I found that again, most k-mers are equally conserved. But Gal4 is much more conserved upstream. And that basically says, wow, I can basically select those motifs that are upstream conserved and then find them. The elephant in the room is that, oh my, there's a lot of motifs that are preferentially conserved downstream. That's kind of cool. It basically says that there's a whole other class of regulatory elements that are acting downstream of transcription, perhaps to terminate transcription, do all kinds of other cool stuff. So we can basically start enumerating. We can basically take these motif seeds, 3, gap 3, and then search them, score them. And then what we can do is actually expand them. We can basically allow for degenerate bases to start filling in the surrounding positions by basically asking for all those conserved instances, what differentiates them, and then fill the unspecified bases surrounding this, and then progressively improve the motif until I get a higher conservation score. And then ultimately, we're going to cluster these motifs. Basically, some 3, gap 3 will basically start here, others will start there. And then putting them all together, we're going to collapse them into a small number of final consensus. Yeah. Yeah, we're going to get at that. So this is one approach. This is the approach that I took in my PhD thesis. So this was published about 15 years ago, gosh. And I feel just as young. I may not be just as young. And then there's another approach that Amos Tanai took. He's now a professor over at the Weizmann Institute, where he basically said, instead of looking at conservation, I'm going to see how often are motifs exchanged with each other. How often do I have one k-mer in one species and a different k-mer in another species? And then these exchange patterns will paint families of k-mers that are frequently exchanged. And that approach actually allowed him to discover motifs in just as easy a fashion, and even to discover the degeneracies of these motifs by walking along this graph. So then your question is, how many of those are experimentally validated? How many can you discover? So the first thing you can do is basically ask how many in fact match known motifs. So there, you know, there's like decades of research on discovering motifs. And, you know, indeed, a large fraction of them match motifs. You could also ask how many of them are novel motifs that actually make biological sense. And then you can use all kinds of techniques for finding biases in those novel motifs. So basically, if we have a bunch of motifs that we don't know the function of, we can start asking, are they near genes that are co-expressed? Are they enriched in, you know, regions that are bound by the same regulator, or regions that are showing the same chromatin state, and so on and so forth. And that allowed us to actually assign putative functions to many more motifs that were not previously known. So this not only captured a lot of the known motifs, it also discovered motifs that were not previously known. So everybody with me here? So basically, just like I can discover motifs that are in the same region based on this iterative sampling, I can discover motifs completely de novo by basically searching for conserved k-mers across the genome and then collapsing these k-mers together using either overlap or these graphs, and then end up with a small set of motifs, which very frequently have in fact recaptured known regulatory motifs. So I did not have the, but gosh. So this is the actual paper. And, you know, here's the slide that's motivating the approach. And then I'll show you now the actual motifs. So this is what was discovered. And, you know, you can basically see for every known motif, that was exactly your question. You basically said, hey, are known motifs conserved? And what you can see here is that ABF1, the very motif that I showed you on the very first slide, in fact showed one of the strongest conservation scores. You can see here the consensus sequence of this, you know, motif here, and it's actually extremely strongly conserved. And then all of these other motifs are in fact very strongly conserved. Gal4 is down here. It's not the strongest, but it's one of the strongest. And you saw that there were all these dots up there that were much more strongly conserved. This is what they are. And then conversely, you can start from discover motifs and then ask, you know, are these previously known? And in fact, the strongest motif is in fact, you know, ABF1 and so on and so forth. So it's kind of cool, right? You can basically see that, you know, you can recover the known motifs and you can start assigning functions to novel motifs. So these are the novel motifs that we found. And, you know, now I'm basically sorting solely based on what was discovered, regardless of whether it was known or not. And then I'm basically saying, what is the category that was the most enriched for that motif? And indeed, the top motif was indeed ABF1, and then REB1, and then some expression cluster, and then some, you know, gene ontology term, and so on and so forth. And then many of those matched known motifs, and then some sequences matched novel motifs. So if this one is mitochondrial downstream, this one is filamentation, this one is switch for variable gap, and so on and so forth. Who thinks this is kind of cool? Awesome, yay. They're so nice. All right, so this is now allowing us to discover these motifs de novo, but what we would like to do also find their individual instances. So this is basically now going from the regulators knowing the motifs to the instances. And to do this, we normally use experimental methods such as ChIP-seq. But what we wanted to do is basically use a computational way of finding them. And then the challenge, of course, in target identification is that, some instances will be perfectly conserved. Other instances will move around, will preserve their function, even though they're not, at the same exact location. And others will simply not be detected because some sequences are just not captured in the other species. So what we need to do is basically figure out a way to account for this uncertainty, to basically search a particular motif instance, such as CTCF, in a bunch of species, and then wherever it occurs, whether there's a gap in the alignment or not, it will basically tell me, yes, this motif is found when I remove all the gaps and I realign sequences locally and I allow movement. And here's the total branch length over which it is conserved, okay? And that we're gonna now transform into a measure of significance. So we're gonna measure the branch length score, simply adding up all the branches over which it's conserved. And then we're gonna be allowing for mutations that preserve the sequence, and we're gonna be allowing for movement of the motif. And if there's some small branches that are missing, it's not a problem. That's gonna be accounted for. So what we'd like to do now is translate this branch length score into a confidence score. And to do this, we need a background model. We need to know how much branch length would I have expected by chance for a motif of that type. So to do that, because motifs vary greatly in their composition, they vary greatly in their length, in their information content, in their dimer composition. So we're going to be searching for how frequently are motifs of that type occur. So this is the, what I would expect for the number of instances that are found at every branch length score. So what I basically do is that if I have a very, very strong branch length score, there's very few instances. And as I reduce the threshold of scoring quality, if you wish, then there are more and more and more instances. But I need to compare this with expectation. And what I can do is basically use motif-specific shuffled control motifs to determine the expected number of instances at every branch length score by chance alone, or due to non-motif conservation. And then I'm going to compute the confidence score as the ratio of the two. The further I go to the right, the higher the fraction of motif instances at that branch length score that come from my true signal. But as I go to lower and lower scores, the noise seems to be increasing. So at a particular cutoff, say, a score of 0.9, I have 75% of my instances that I would expect at that threshold coming from true signal and only 25% coming from noise. So at that branch length score, I will translate it to 75% confidence. Everybody with me on this translation? Raise your hands if you're following. Awesome. Great. So that's how we're going to produce control motifs, hundreds of shuffles, and then filter the motifs, sort control motifs based on the similarity of known motifs, and then cluster them, and then end up with a family of control motifs that we're going to be using for testing. We're going to compute enrichments in the same way by randomizing our regions. And then when we do that, what we basically do is that we find that as I increase the threshold of confidence, I end up with transcription factor motifs selecting instances of those motifs in promoter regions. But for microRNA motifs, I end up selecting instances in 3' UTRs. So that basically means that I am, actually selecting functional instances because of this bias. You can see that initially they all start at the same frequency, and that's simply what fraction of the genome is covered by each of these classes. But as I increase for TF motifs, it's selecting promoters. For microRNAs, it's selecting 3' UTRs because they're different from each other. Says that it captured actually something biological about it. And then we can actually start validating these motif instances. So we found a bunch of instances. How do we validate that they're real? What we can do is for microRNA motifs, ask if they're biased in their strand, because an RNA molecule has only one strand. So if I search the opposite strand, you'd expect to not work. And indeed, for TF motifs, both strands seem to work equally well. But for microRNA motifs, if I increase the confidence threshold, I end up finding them preferentially in only one strand. I can also ask how often are they conserved in other species, how often are they bound by chromatin immunoprecipitation experiments, and so on and so forth. And what we find is that the validation rate increases with the confidence. As I increase the confidence, more and more of them do validate. So that's basically allowing me to now say that indeed this approach truly finds, de novo these true instances. So that's basically now discovering motifs yet in another way, and then identifying their instances using evolutionary conservation. What I'd like to do now is switch gears again and talk about yet a third approach for dissecting the regulatory potential of these regions. And this is now focusing on the specific instances of these regions. Okay. So as we heard in the epigenomic lecture, there are many, many different regions that have signatures of enhancers, signatures of promoters, like DNA accessibility, like H3K27 acetylation, H3K4 monomethylation, H3K4 trimethylation. So we can use these epigenomic signatures to predict candidate regulatory regions. But how do I know that these candidate regulatory regions are actually functional? So what I need to do is see if I take that chunk of DNA and I put it in a reporter construct upstream of a luciferase gene, which basically shines green. Then how often do I see that the cell that receives this construct, where luciferase is downstream of my predicted enhancer, how often does it actually drive expression? How often does it actually function as an enhancer? Can everybody see how this construct works? I'm going to basically take a piece of DNA that has the sequence that I want to test, and then ask if that, when I put that sequence in front of a reporter gene, that reporter gene gets turned on. Who's with me on this one? Raise your hands. Awesome, great. So when Weisel et al. Did this, they tested thousands of elements, and they found that only about a third actually function, or about a half function well, and truly drive enhancer activity. So how do we test this? So, you could do 3,000 experiments, or you could develop new technologies for doing this systematically. And the technology that I want to talk about today is this massively parallel reporter assays technology. So what that allows us to do is, instead of putting a single enhancer, I'm going to put 10,000 enhancers in one experiment. How? By synthesizing them in a microarray. So I can order a microarray. They will generate that microarray. I can cleave off the little fragments from the microarray, and I will end up with 10,000 sequences. Everybody with me on this? How I can synthesize them? So just like I can print a microarray, I can just print all of these as different spots in the microarray. And there are 145 base pair spots. Okay? I've now synthesized them. Then I insert each of them into a different reporter construct, and I add a barcode. And when I add a barcode, I can then PCR amplify the common sequence, which is flanking this barcode, and simply count barcodes. And when I count barcodes, I will know how often did I observe a barcode that corresponded to each of the enhancer regions that I'm testing. Who's with me on this one? Raise your hands. Awesome. So when I do this systematically, I can basically end up with, many different kinds of approaches, MPRA, ChIP-seq, STAR-seq, etc., for basically testing tens of thousands of these elements at once. So you have to deal with a lot of experimental artifacts, indeed. Yes, so you have to deal with that. So here's this, approach specifically developed by Tarsha Mikkelsen that we were involved in, that basically synthesizing all of these different sequences, inserting them, and then using a different barcode downstream of each of them, transfecting them into a cell and into millions of cells, and then testing how often did I see each of the barcodes. And you can use this for example, testing what is the effect of a single nucleotide polymorphism on the expression. And, you know, this allows you to now evaluate 10,000 variants in a single experiment. Alternatively, what I could do is use a different version with a single nucleotide change at every one position of the same construct. So here I'm taking the CRE promoter, and here I'm taking the INF beta promoter. And for every single one of them, we can basically ask, what is the effect of testing that entire sequence minus this particular nucleotide, where I'm going to now change every position into an A, into a C, into a G, or into a T. And then what it shows me is that the activity changes for these particular locations. And those particular locations correspond to exactly where the known motifs were. So by basically using 10,000 enhancer constructs in one experiment, I can basically evaluate the impact of mutating every single nucleotide. Who thinks this is kind of cool? Awesome. So I can do this here, I can do this in another sequence, every single time inferring this. But the challenge is that we need hundreds of constructs for testing a single region. Can we test thousands of regions jointly? So the answer is yes. What we could do is use our motif predictions to test thousands of regions, perturbing only that one motif in every one of these regions. Sounds good, right? So what we're going to do is use the wild type sequence, shuffle it, cut it out, change a single nucleotide, and so on and so forth. Okay? And then we're going to build a different reporter construct for each of those. How do we select those? We're basically looking for the expression of the regulator, the activity in the specific cell type, and then we're using that to basically predict who are the activators and who are the repressors of every single cell type. And when we do that, we basically find that if I take a predicted activator like HNF4 and its motif, then when I test the wild type motif in the correct cell type, I find that indeed it matches and it shows expression very well. If I scramble, remove, or maximally decrease that motif, then it abolishes reporter activity. If I change it ever so slightly, sometimes I get a better and sometimes I get a worse score, but I maintain activity. If I do a random change that perturbs a motif, I lose it. So every single measurement here is a different barcode used for the same construct. We're doing a lot of replicates, enabling us to now infer the activity of every one of those. Okay? So I can take a step further, and instead of testing a single sequence, I can offset things by 50 nucleotides at a time. Okay? And when I do that, I basically find that sometimes, as I get closer to the predicted site, things increase in value. And in fact, I find regions where, you know, none of these offsets work, and suddenly this offset and that offset work very well, and then this offset doesn't work anymore. And when I ask what is the difference between them, there's a 30 base pair segment which is unique to the regions that work the best, and that segment indeed contains the motif for the HNF4 transcription factor that, you know, is active in that cell type. So that basically means that by tiling regions and offsetting them, I can actually figure out, perhaps in high resolution, where the motifs are. Who's with me on this concept? Awesome. So why do 50 nucleotide increments? Let's instead do 5 nucleotide increments. So now we're going to be testing these regions at 5 base pair offsets and inferring computationally from these offsets what is the likely activity for every base pair interval in the genome. And when I do that, I find regions that just simply don't work, and suddenly, boom, work. And the computational algorithm basically says, aha, the specific sequences that work are the ones here. And it allows us to now start predicting in high resolution where are the driver nucleotides for every one of those positions. Everybody with me on this? The last thing that I'm going to talk about is a little crazy. We basically said, why test 15,000 regions at a time if you could test 7 million fragments at a time. So what we did is, instead of generating a microarray, we simply cut out accessible regions using an attack-seq experiment, the same thing that we learned in the epigenomic lecture. And then you're cutting out all of the DNA fragments that are coming from accessible regions, and you're inserting them not upstream of your construct, but downstream of the construct. That means that when they drive expression, they will drive their own transcription. And that also means that I can actually use them as their own barcodes. Everybody with me on this? So they're self-transcribing constructs using a technique called StarSeq that Alex Stark in his lab developed. He was previously a postdoc in my lab. So using this, we basically tiled all of these accessible regions, and we're able to, in high resolution, infer exactly where the activity is coming from using another deconvolution algorithm called Sharpr that can work at any variable offset. So this is where I'll stop, and basically recap by saying that motifs are at the core of all of these regulatory genomic assays, and there are techniques for finding those based on their overrepresentation, for discovering them de novo based on their evolutionary conservation, for finding their individual instances using this branch length score, and then for de novo dissecting in high resolution these regulatory regions.